Hello everyone, welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with the motto to make plant pathology easy. We are here today at day 3 of our ASRB NET 2025 revision series. So yesterday we covered the first half of the general plant pathology and today we will begin with the second half of the same general plant pathology. So the concept remains the same. After each question, I will pause for 2 to 3 seconds. Just look at the options and note down whichever one clicks your mind. Alright then, without any delay, let's begin with day 3. So yesterday we have done till question number 127. Today we will move forward starting with question 128. So Southern blot technique is discovered by E.M. Southern and Northern blot technique is discovered by option A. Davis et al. Option B. Elwin et al. Option C. Breyer and Staxwees et al. Option D. Dewey et al. The answer is option B. Question 129. Wound tumor virus is under of leafhopper transmitting virus and it is detected by A. PCR, B. Indirect ELISA, C. Direct ELISA or D. Fluorescent antibody technique. Answer is option D. Question 130. ELISA plate is made up of option A. Iron, option B. Plastic, option C. Polystyrene, option D. Aluminium. Answer is option C. Next question 131. Sun scald of tomato is due to high temperature, low temperature, low oxygen level or low light intensity. This is due to high temperature. Question 132. Billet belong to the fungicide group option A. Carbendazim, B. Benzimidazole, C. Tridamorph and D. Oxethin. This is option B. Question 133. Component of Bordeaux mixture are copper sulfate plus hydrated lime, copper sulfate plus sodium chloride, copper carbonate plus hydrated lime or copper oxychloride and lime. Answer is copper sulfate and hydrated lime. 134. PCR can amplify RNA, protein, DNA or both DNA and RNA. Both RNA and DNA same. Which one? Answer is option C, DNA. So what will happen in case of RNA viruses or the pathogen whichever is having RNA as their genetic material? So in that case, first RNA has to be converted into cDNA, means complementary DNA, and after that the DNA will be amplified by the PCR. Okay, next question 135. Inspection certification are successful for controlling of option A. Smut disease of cereals, B. Viral diseases of potatoes, C. Bacterial diseases of legumes or D. Viral diseases of citrus. The answer is option B. 136. For a pathogen, IRC is more than one. It can create an epidemic, endemic, pandemic or sporadic. Answer is option A. 137. CGA 245704 is a elicitor, viricide, plant defense activator or bactericide? Answer is C. Plant defense activator. Next question. Compound interest diseases over time produces which type of curve? Linear curve, parabola curve, hyperbola curve or sigmoid curve? The answer is sigmoid curve. So, do you know compound interest diseases are actually what? They are the polycyclic diseases. Okay. Question 139. Alcinophausity causes citrus cap is belong to A. Kingdom fungi, B. Kingdom protozoa, C. Kingdom chromista or D. Kingdom plantae? Answer is Kingdom fungi. Question 140. Low volume sprayer contain water about 25 liter, 100 liter, 500 liter or 700 liter? The answer is B. 141. Fungicide specific to rust. Is it Vitavax or carbendazim, metalaxyl or Plantavax? Yes, the answer is what? A or D? See, option B and C are not relevant. When the question is asked rust, then we normally get confused between Vitavax and Plantavax. So one of the answer is for rust, whereas the other will be for smut. So we can remember one, not to get confused. Okay, so for rust, the fungicide is Plantavax. So Vitavax is used against smut diseases, right? For rust, Plantavax. For smut, Vitavax. Coming to question 142. Fungicides specific towards oomycetes fungi are under Vitavax, carbendazim, metalaxyl or Plantavax? Answer is option C. Yes, when they are asking about oomycetes fungi, then this Vitavax and Plantavax will not be the answer, right? So for oomycetes, it's metalaxyl. If we can remember it directly, then it's fine, or else we can do elimination. Question 143. Which one is not a protein? Zymase, macrozyme, DNA polymerase or ribozyme? The answer is option D. 144. Generally, high mutation rate is not visualized in diploid due to option A recessive, B dominance, C aggressive or D tolerance? Answer is option A. 145. High mutation rate is tolerant by option A haploid, B polyploid, C diploid or D aneuploid? Answer is option C. 146. Non-pathogenic microorganisms when entered into plant cell is produced some clogging exudates which are known as vesicle, papilla, penetration peak or protuberance. Answer is option D. Moving to 147. Express of resistance in plant disease reaction from susceptible appropriate treatment is called functional resistance, induced resistance, structural resistance or synthesis resistance. Answer is option B. 148. Which is or are the effective biological control agents? A. Agrobacterium tumefaciens, Gliocladium virens and Trichoderma species, C. Bacillus thuringiensis, or all of the above. The answer is all the above. Okay. Question 149. Cytoplasmic inheritance is governed by plasmid DNA, nuclear DNA, extranuclear DNA, or all the above. Answer is extranuclear DNA. 150. Chitinase or beta-1,3 chitinase is a phytoalexin, elicitor, pea protein, and or enzyme. Answer is C, PR protein. But there are 19 PR proteins are there, out of which the function of 17 PR proteins are known, the rest 2 are unknown. So chitinase, beta-1,3 chitinase is which type of PR protein? That is something you should learn, like the functions of different PR proteins. Okay. Question 151. Cleaning solution for glasswares is chemically known as formalin, mercury chloride, sodium chloride, or potassium dichromate. Answer is option D. Question 152. Hybridoma technology is a new biotechnological approach for commercial production of monoclonal antibodies, interferon antibodies, or alcohol. It is monoclonal antibodies. Question 153. Which one of the following organisms can serve as a biofertilizer for rice crop? Rhizobium, mycorrhizal fungus, glomus species, Trichoderma viride, or blue-green algae? The answer is option D. 154. Insect-resistant cotton plants have been genetically engineered by inserting a gene from a or an virus, bacterium, insect, or is it plant? Answer is bacterium. But which bacteria? Can anybody tell what is the natural genetic engineer? The name is Agrobacterium. Okay. Question 155. During breeding for disease resistance, total number of bag cross made in the range of 5 to 6, or it's 7 to 8, 9 to 10, or 10 to 11. Answer is 5 to 6. 156. The first commercially available biocontrol agent or plant pathogen was Trichoderma hercianum, Pseudomonas fluorescens, Peneophora giganteum, and or Bacillus subtilis. The answer is option C, Peneophora giganteum. 157. The first mycoherbicide registered in the US Environmental Protection Agency is Coligo, Divine, Cast, or Libo 2. The answer is option B. So again there will be this confusion between question number 157 and 158, because the first mycoherbicide was Divine, but which one was the first commercial mycoherbicide? If the question is asked commercial mycoherbicide, then it will not be divine. Okay, in that case the answer will be which one? D, Coligo. Okay. 159. Crissic phase, common symptom found in, it's very easy, do it fast. Option A, late blight of potato. Option B, black gum disease of cotton. Option C, BLB. Option D, muco disease of banana. Answer is BLB, bacterial leaf blight of rice. 160. DAPI staining is a practical method for detection of which one group of organism in laboratory? Bacteria, fungi, viruses or phytoplasma? Answer is yes, phytoplasma. 161. The only sugarcane disease resulting in increase in sugar content. Look, I love this question. Okay, which disease of sugarcane increases the sugarcane rather than harming it, or harming it in a sense, if it increases the sugar content, so it is benefiting the farmer rather than harming the economic or the economic loss. Coming to the answer, okay, whether it's grassy stunt or red trot or ratoon stunt or smut, the answer is ratoon stunting. Okay, so ratoon stunting increases the sugar content of sugarcane plant crop. Okay, coming to 162. Which of the following pathogen of potato causes 100-fold increase in auxin content as compared to healthy plants? Option A, Ralstonia solanacearum. Option B, Phytophthora infestans. C, potato virus Y, or D, Rhizoctonia solani. Answer is potato virus Y. Question 163. The level of horizontal resistance tends to decline as a result of breeding for vertical resistance is known as A, killing reaction, B, vertifolia effect, C, operant concept, or D, hypovirulence phenomena. Which one? The answer is vertifolia effect. Question 164. A subpopulation of the host in which all individuals have a specified character of resistance in common is known as? The options. Option A is pathotype, option B is vertifolia effect, option C is pathotype, or option D, none of the above. So the answer is option C. Okay, now we have 4 questions in which we have to memorize the values. Okay, so LD50 value of carbendazim is 6400 mg per kg, 3033 mg per kg, 2000 mg per kg, or 660 mg per kg body weight. Answer is option A. 166. LD50 value of hinosin 20 mg per kg, 100 mg per kg, 2000 mg per kg, or 6400 mg per kg body weight. Answer is 100. I'm saying the answers because I guess these values are not easily remembered. Question 167. LD50 value for carbendazim is 100, 2000, 3200, or 5770 mg per kg body weight. The answer is 3200. Yes. 168. LD50 value of cycloheximide is 2.5, 100, 3200, or 15,000 mg per kg. Answer is what? 2.5. Question 169. Paddy straw mushroom grow best at a temperature range of 18 to 25, 25 to 30, 30 to 35 or 35 to 40. The answer is C. 170. Pore size of filter used for laminar flow air chamber is 2 micrometer, 1.75 micrometer, 1.25 micrometer or 0.3 micrometer. The answer is D. 171. The mechanisms of biological control of plant pathogens is or are competition, induction of plant defense, parasitism or all the above. Answer is all the above. 172. PR proteins are synthesized principally in membrane-bound polysome and mRNAs, cell wall, ribosome or none the above. Answer is option A. 173. PR proteins accumulate in the apoplast and vacuole, chloroplast, ribosome or none of the above. Answer is apoplast and vacuole. So PR proteins are synthesized in membrane-bound polysomes and mRNAs and they accumulate in apoplast and vacuole. Okay, remember the difference when they are asking synthesize and when they are asking about accumulation. Coming to 174. Indian childhood cirrhosis, a disease of children, is due to the consumption of peanut contaminated with bacteria, virus, aflatoxin or algae. Yes, it's aflatoxin. Aflatoxin is mostly found in groundnut caused by Aspergillus flavus mostly, but one more Aspergillus species is also there, like Aspergillus flavus and Aspergillus parasiticus, but mostly Aspergillus flavus. If you have to choose one, then it should be flavus. Question 175. Turkey eggs disease was due to a virus, bacteria, mycoplasma or aflatoxin. Yes, it's again aflatoxin. Question 176. The optimum RH means relative humidity and temperature for aflatoxins productions are option A 40 to 50% and 15 to 20 degrees Celsius, option B 80 to 90% and 25 to 30 degrees Celsius, option C, this will be C, 60 to 70% and 20 to 25 degrees Celsius, option D, 70 to 80% and 10 to 15 degrees Celsius. The answer is option B. 177. Which of the following pathogens is a lower sugar pathogen? Alternaria, Cladosporium, Erysipeus or Buccinia? Yes, it's Alternaria. Because see, low sugar pathogens are the necrotrophs. Whenever they will ask the high sugar pathogens, okay, the next question, see, which of the following pathogens are high sugar pathogens? Whenever they will ask high sugar pathogen, means the pathogen should be obligate. In the option, just go and search for the obligate pathogens. So in high sugar pathogen case, Erysipeus will be the answer. Whereas in low sugar pathogen, see Erysipeus, Buccinia, Cladosporium all are obligate. Whereas Alternaria solani, this is a necrotroph. So in this case, Alternaria was the answer. In high sugar pathogen, Erysipeus will be the answer. Okay. Question 139. I just told you this. Low sugar pathogens are generally biotroph, necrotroph, obligate or symbiotic. Just now I told you. Just do it quickly. So low sugar are necrotrophs, whereas high sugar pathogens are biotrophs. Okay. Okay. 181. Low sugar pathogens attack more severely in younger leaves, older leaves, new leaves or fallen leaves. Answer is older leaves. So they have not asked about the high sugar pathogen, but what will be the answer then? High sugar pathogen attack more severely on which type of leaves? Younger leaves. If the high sugar pathogen, they are generally biotrophs, so they generally target the new leaves or the younger leaves. Okay, the low sugar pathogens are necrotrophs, so they severely attack the older leaves. Okay, coming to 182. Agar-agar is composed of glucose units, amino acids, D-galactopyranose units or D-fructose units. Answer is C. Galactopyranose. 183. For culture media preparation, concentration of Japanese agar-agar required. For India we know, but they have asked for Japan and New Zealand. So for Japan, the agar-agar requirement is 4%. Okay. And for New Zealand, the requirement for the culture media of agar-agar is 1.2%. 185. Agar-agar is it a protein, long chain of polysaccharide, lipid or glycoprotein? It is long chain polysaccharide. 186. Gelatin is a protein, or I guess it will be protein. Same. Gelatin, is it a protein or long chain polysaccharide or lipid or glycoprotein? So gelatin is a protein. Question 187. The water-soluble ingredient peptone used in culture media preparation is obtained from sea algae, seaweed, lean meat or other protein material or none of the above. The answer is option C. 188. Medium containing gelatin is sterilized by autoclave, pressure cooker, radiation or fractional method. Answer is option D. 189. Meat extract is commercially known as brewer's yeast, peptone, lab lemco or baker's yeast. The answer is C. Coming to question number 190. For measurement and adjustment of pH of culture media, the most accurate method is electrometric methods, indirect dye method, colorimetric method or pH paper method. The answer is electrometric method. Question 191. An example of simple interest disease is lead blight of potato, soil-borne smut disease, stem rust of wheat or none the above. It's soil-borne diseases. Be it smut or any other disease, soil-borne diseases are normally monocyclic, and monocyclic diseases are the simple interest disease. Okay. 192. An example of compound interest disease is, so if monocyclic are simple interest, so compound interest polycyclic. It's told earlier. Options are soil-borne smut diseases, vascular wilt disease, stem rust of wheat or none. Answer is option C, stem rust. Okay. Question 193. The toxin fusaric acid is lethal to host tissues at a concentration of
10 to the power 4, 10 to the power minus 3, this is minus 4, minus 2, or 10 to the power minus 1. Answer is 10 to the power minus 4. 194. Which of the following fungicides is commonly known as Kittleson's killer? We have discussed it in the history section as far as I remember. Kittleson's killer is Captain. Oh, I did not say the options. Sorry. Okay, so Kittleson's killer is Captain. Next, see in the next question in 195, the answer was already written. It's not my fault. See, Kittleson's killer in bracket Captain was introduced in 1934, 1952, 1966, or it's 1971. Which one? Option B, 1952. 196. Time taken by simple interest degree to produce a disease progress curve. It's over different weeks, over different months, over fortnight interval from seed germination to maintain of the crop in one season or one disease season. Answer is one disease season. 197. The highly active oxygen radical are thought to be released by mitochondria, nucleus, cell wall, or cell plasma membrane. Answer is cell plasma membrane. 198. Which one of the following is a wound hormone? Bisatin, chromatin, rishitin, or osmotin? It's option B. 199. The phenomenon of horizontal gene transfer is found in host pathogen. Relation of Agrobacterium, Bacillus, Clavibacter, or Erwina. Answer is horizontal gene transfer. Act as a vector. Answer is Agrobacterium, option A. 200. Question 200. Simple interest disease over time produces which type of curve? Sigmoid, saturation, hyperbolic, or straight? The answer is option C, hyperbolic curve. 201. Juvenile or early growth resistance found in diseases caused by Botrytis, Penicillium, Monilinia. Option B, Pythium, Taffarina. Option C, Plasmapora or Puccinia, or option D, bacterial blights or viral infections. So the answer is option A. 202. Slow epiphytics are found in which type of crops? Annual, perennial, vegetable, or cereal? Answer is perennial crops. Remember, for slow epiphytic, perennial crops. For rapid epiphytics, then it will be annual crops, yes. Question 204. Which pathogen groups are under compound interest disease? By now, we have understood compound interest means polycyclic disease. So, the options are Cercospora and Phylosticta. Option B, Phytophthora investans and Puccinia graminis. Option C, Syncytium endobioticum and Streptomyces cabis. Option D, none of the above. Answer is yes, Phytophthora and Puccinia. In last question, we have seen a polycyclic disease, compound interest were late blight of potato and stem rust. So yes, option B, Phytophthora and Puccinia. 205. Polycyclic disease produce disease progress curve of saturation, straight, hyperbolic, or sigmoid? Answer is sigmoid. Whereas for monocyclic disease, they produce which type of curve? The options are same, but the answer will be different. Answer is yes, saturation. Now you must be wondering, there was one question of same type. See, simple interest disease over time. So you must be asking ki simple interest and monocyclic diseases are same. So why in this case it's saturation and in that case it is hyperbolic, right? So in general, disease progress curve, it's saturation for monocyclic and sigmoid for polycyclic. But when it is measured over time, it produces a hyperbolic curve. Okay. 207. Which of the following element is contributed to the disease and drought resistant in plants? Nitrogen, calcium, potash or iron? Answer is potash. 208. The flagellate protozoa Phytomonas francae causing empty root of cassava are found in xylem, phloem, laticifer or leaves. Answer is C. 209. Some plants synthesize some antimicrobial compounds known as phytoncide, phytoalexin, siderophores or elicitor. Answer is yes, phytoalexins. Okay. 210. For preparation of Bordeaux mixture, the composition new in a ratio of 555. Okay, there 5 lb, 5 liter. Here 5 kg, 5 kg and 5. Option C is 5 lb, 5 lp or 50 gallon water, or 5 kg lime, 5 kg lime or 55 gallon water. Which one will be the answer? Look at the questions, options. It's a little bit confusing, but the answer will be option C. 211. Which is a sterol inhibitor? Calixin, oxethin, metalaxyl or tridamethon? Answer is D, tridamethon. 212. The process to application of fungicide through irrigation is fungi irrigation or fungigation or fun irrigation or none of the above? Option is B. Fungicide, the application of fungicide, that's why fungigation. If the question was asked application of chemicals, then chemication, or the application of what it's called fertilizer, then fertigation. Okay, similarly. Question 213. The fungicide used for rescue treatment. It's bevacizine, henocin, tridamethon or Bordeaux mixture. Answer is tridamethon. Question 114. Cultivars of PGNP resistance of to wilt have high content of option A cysteine, option B methionine, option C valine, option D histidine. The answer is option A. 215. Photoreaction phenomenon is related to A enzyme, B toxin, C vitamins, D nucleic acid. The answer is nucleic acid. 216. Nectaria cinnabarina causes infection of fruit trees by penetrating through stomata, wounds, hydatodes or roots. Answer is wounds. 217. Resolution of a microscope is limited by wavelength of light, size of the object or objective, power of lens or numerical aperture. Answer is wavelength. 218. The epidemic of apple scab occurred in Kashmir Valley in 1943, 1960, 1973 or it's 1979. The answer is option C. 219. The resolving power of light microscope is 6 micrometer, 3 micrometer, 0.6 micrometer or 0.3 micrometer? The answer is option D. 220. Effect of one or more environmental factors which make a plant vulnerable to attack by pathogen is known as symbiosis, predisposition, induction, or susceptible factor. Answer is B. 221. In antigen-antibody reaction, precipitation bands are formed in tube precipitation test, ELISA test, slide agglutination test, or none of the above. Answer is tube precipitation test. 222. Indian type culture collection center is located at Mumbai, Kolkata, New Delhi, or Hyderabad. The answer is New Delhi. 223. Cheese are ripened with species Aspergillus, Penicillium, Mucor, or Saccharomyces. Answer is Penicillium. 224. The book entitled Fundamentals of Mycology written by Burnett, Bessey, Gorman, or Alexopoulos. Answer is option A. 225. Apothecia are found in Puccinia, Pizizia, Penicillium, or Pyrenula. Answer is option B. Question 226. Which of the following fungi is known as kerosene oil fungus? Option A, Rhizopus. Option B, Epsidia. Option C, Amorphothera. Option D, Zygophyncus. The answer is option C. 227. DAPI stain is used to detect fungi, virus, bacteria, or protozoa. Answer is bacteria. 228. Fatty acid analysis technique is used to detect fungi, virus, nematodes, or bacteria. Yes, again it's bacteria. 229. Page technique is used to detect fungi, virus and viroids, protozoa, or nematodes. The answer is option B. 230. Baiting technique is used to detect fungi, virus, nematode, or protozoa. Yes, it's fungi. 231. Leaf deep technique is used to detect fungi, virus, nematode, protozoa. Answer is virus. 232. PCR technique is used to detect fungi, virus, nematode, all. Yes, it's all of the above. Molecular, which is PCR, can be used everywhere. 233. Everywhere in a sense where obviously it amplifies the DNA. So genetic material DNA should be there, or RNA at least. Okay, 233. Agdia multi-well ELISA kit is used for the detection of fungi, nematode, FVB, or protozoa. The answer is option C. 234. The cells surrounded by a wall made up of mucopeptide are found only in the case of fungi, bacteria, phytoplasma, or protozoa. The answer is bacteria. Okay, 235. Which of the following organisms may be a connecting link between pro and eukaryotes? Pro means prokaryotes and eukaryotes. Pilomixa, Vibrio, Pseudomonas, or Salmonella? The answer is Pleomixa. 236. Metalaxyl was discovered in 1952, 1966, 1977 or 1980? Answer is 1977. 237. The term epidemic means among the people, great loss, catastrophic or plant disease. Answer is among the people. It means a sudden disease outbreak that affects a large number of people in a particular region, community or population. 238. The word smut means cottony-like, smooth powder, white powder or sooty or charcoal-like powder. Answer is option D. 239. The condition in which the change of color to red, purple or orange is known as albinism, chromosis, chlorosis or etiolation. Answer is chromosis. 240. The abnormal increase in size of a plant organ due to increase in number of cells is known as hypertrophy, hyperplasia, okay, scat. These two options look same. So, okay, you can choose either this or this. Fine. Hyperplasia. 241. Organomercurial fungicides were first discovered by Tisdale and Williams, Mason, Bewley or Robertson. It's Tisdale and Williams. 242. Frost ban A is a product of Pseudomonas agrobacterium, erwina or Pseudomonas fluorescens. Means Pseudomonas syringae or Pseudomonas fluorescens. Yes. Yes, it's syringae, not fluorescence. 243. Hypervirulence was discovered in blister blight of tea, chestnut blight, Dutch elm disease or cedar apple rust. Yesterday we have covered this question. Not the specific question, but the type of question. So the answer is chestnut blight. 244. Sexual stage is also known as teleomorph, anamorph, benomorph or sexomorph. Answer is teleomorph. The asexual stage, anamorph. Sexual stage, teleomorph. Okay. 245. The term quarantine literally means obstruct, disease control, 40 days period or restriction. It is 40 days period, 40, not 14. 246. Question number 246. A normally white or colored tissue that develops chloroplast and becomes green is known as virescent, fasciation, scotch or chromosis. Answer is option A. 247. The safest and most effective method of plant disease control is chemical, use of resistant variety, biological or cross protection. Answer is resistant variety. 248. What disease of potato in Darjeeling district? It is pandemic, epidemic, sporadic or endemic. Means pandemic, epidemic, sporadic or endemic. Answer is endemic because it is restricted to Darjeeling, this area only. 249. Late blight of potato pathogen can overwinter in insects, water, plant debris of potato or soil. Answer is option C. 250. Peach leaf curl is caused by virus, fungus, phytoplasma or bacteria. Answer is B. Fungus. Which fungus? Taphrina deformans. 251. Based on membership, the Indian Phytopathological Society is the first largest phytopathological society, second, third or fourth? This is second largest phytopathological society. Then which one will be the first? If I'm not wrong, then the first will be the American Phytopathological Society based on membership. But then which one will be the first? The first is Netherlands Phytopathological Society. Okay. Okay, see 252, the largest phytopathological society in the world. It is American, Indian, Japan or Netherlands. The answer is A, American. So three things. For India, this is Indian Phytopathological Society. So in overall, this comes second. But according to the size, the first one comes American Phytopathological Society. Whereas the oldest and the fastest, the oldest and the first Phytopathological Society was Netherlands Society of Plant Pathology. Okay, these three things we need to remember. So yes, I also have attached the answers. Take a screenshot of this and this. As far as came to my knowledge, question number 163, it will not be option A, it will be option B. Okay. Okay. So any other changes that you witness, please let me know and let others know as well. No problem. You can share anything and everything in the comment or let me know if I am doing something wrong or do I need to change any way of explanation or any method that I need to change. Everything is open to discussion. So without further ado, I'll take a leave for today and we will come back tomorrow with the next part. I guess tomorrow we will be starting mycology section. Okay, so be ready. Till then, keep learning, keep growing. See ya.